everyone, and welcome to today's webinar, The Powers of Odin, Comprehensive Cellular Characterization. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Biolog. To learn more, visit www.biolog.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box to the left and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Dina Finan, Senior Product Manager at Biolog. For a complete biography on our speaker, please visit the Biography tab on the top of your screen. Dina, you may now begin your presentation.
Thank you for the introduction, and thank you everyone for joining us today. I'm really excited to talk to you about the new instrument we've recently launched at Biolog called Odin, and hopefully get you excited as well, and maybe learn something new about what the powers of comprehensive cellular characterization can do for you. So Biolog and our core technology has been around for a pretty long time, nearly 40 years. We have instruments installed and being used all around the world on 6 continents, with about 5,000 different publications from researchers citing the use of our kits and our products for a lot of different interesting applications. And our technology all really focuses on understanding the metabolism of cells, and then using that understanding to characterize cells' phenotypes. So, what different stuff cells are able to eat or survive on, and how that affects their behavior. And this core technology can be used for many different applications. For example, the metabolic fingerprint that's unique to each organism or cell type can actually be used to accurately identify organisms that are isolated from the environment. And you can use that for QC and environmental monitoring. You can also use the phenotypic profile to help improve your fermentation or bioprocessing to improve the yield of a product you're trying to make from cells, for example. And just to give a sort of big picture, macroscopic example of phenotypes and how, organisms are what they eat, but also just a nice example where genomic methods don't always give you the full picture of what's happening in an organism. So I really like this hydrangea plant. It can produce flowers in many different color varieties that range from pink to purple to blue. Some of the shades are shown here, but that actually has nothing to do with genetics at all. So all of the flowers are producing the exact same pigment molecule. And it turns out that whether the flowers bloom pink or blue is determined by the availability of aluminum and the soil pH. So those two things are actually related to each other because the pH of the soil will determine how much aluminum is available to the plant because it's more soluble in acidic soil. So at low pH you get blue flowers. In neutral soil you get purple or a mix of blue and pink like shown in this picture. And then in more alkaline soils, you get pink flowers. So in this particular case, there's not really a clear purpose that this serves in the plant other than, a convenient pH test for your soil. So is there an evolutionary advantage to being pink or blue? I have no idea. But if you're trying to grow these flowers for a particular market, it would be super useful to know exactly how to modulate their environment to give the result you want. But in many cases, obviously, phenotypic changes that respond to the environment are really crucial for an organism's survival. And so, as I said, all of our technology really revolves around watching what a microbe or a cell can take in to eat, and then how efficiently it can turn that into fuel, and then whether it uses that fuel for respiration or for growth. And the food sources in this case are really chemical substrates that are made of these basic building blocks. And I'm sure that you know that breaking these macromolecules down is controlled by enzymes, And many of those enzymes need cofactors like NADPH to be able to turn over their substrates. And so for our chemistry, we add a redox reporter dye that's cell permeable and colorless when it's oxidized. And then when it gets reduced during these metabolic processes, it turns purple irreversibly. And then we measure this purple color to be able to answer all of these different questions. So for example, what are the microbes eating? And we do this by putting them in a very minimal media and then giving them access to only one substrate at a time in each well of a plate. We can also say how much of the food source can they eat, and we do this by making quantitative measurements of dye reduction. And then how fast are they eating the food source? And so this is done because we're not just taking endpoint reads, we're measuring kinetically to be able to get a lot of data about many substrates all at the same time. And so first, to show an example of an actual research study that used biolog technology to learn something new and important about a microorganism, I wanted to turn to the wheat plant. So this picture is showing a wheat crop that's infected with a fungal disease called Bifusarium, which infects the heads of the plants. You can kind of see that here. This fungi also produces these mycotoxins, and it's one of the major diseases of grain crops in general that's responsible for really significant losses in grain all around the world. So at high concentration of these mycotoxins, this means that the grain that's contaminated cannot be sold or used for food or animal feed. And even at lower concentrations, these toxins can be toxic to both animals and humans. They can cause a really wide range of negative effects. They can have, bad effects on animals' immune systems, on their nervous systems, and other things. And so these are, tightly regulated for how much toxins can be in grains. And we don't fully understand why or when fungi produce these toxins, but people have made isolates that are unable to produce them, and those do show reduced aggressiveness towards wheat. So you can imagine, it makes sense that it's advantageous to weaken your host. You weaken their immune system, and so then you can proliferate more easily if you're a fungus. So it's been known for a while that, producing these toxins by the fungus really depends on the environment that they're growing in. And so I told you that Biolog has been around for a while, and indeed, here's a publication from quite a few years ago that was one of the first reports of specific conditions that turn on the synthesis of this mycotoxin in this exact Fusarium fungus. And I included a picture of the fungus here because even though they do a lot of harm, they're also really amazing organisms, and sometimes they're kind of beautiful to look at. So in this paper, the researchers used some Biolog plates and a GFP reporter strain to look at the effects of different culture conditions on toxin production. And we have these arrays of plates with sort of pre-selected conditions, and I'll talk more about them in a minute, but that makes it really easy and fast to screen through a range of conditions. So on this particular plate, they were able to look at a lot of different nitrogen sources, and we have combinations at high pH and low pH, and they found really strikingly this huge difference in the amount of toxin produced at low pH, 4.5, with L-ornithine compared to a different nitrogen source like homoserine. You can see the two arrows. So they saw that the low pH and some specific amines actually acted synergistically on the biosynthesis of the toxin. So this is a really nice example of how fast it is with these Biolog plates and instrumentation to be able to screen through many conditions at once and quickly find the conditions that lead to something that you're looking for. In this case, the phenotype was increased toxin. And of course, being able to understand the environmental conditions that lead to increased toxin production is very important to be able to better manage the storage of these grains and to come up with new strategies to control the harm that they cause. So this is just one example of many that are out in the literature. And with that, I'm really excited to introduce you to Odin, which is our newest platform for being able to carry out all of these experiments that I'm describing. So Odin can incubate and measure all of the microplates that Biolog produces and sells. And with Odin, you can measure the formation of that redox dye to look at metabolism of cells. And you can also look at cell growth with more traditional optical density measurements. So we actually measure every plate at 2 wavelengths, either 490 or 590 nanometers, as well as 740 nanometers. And Odin can handle any throughput of experiment. So you can run just 1 plate all the way to 50 plates at a time. And multiple users can actually run plates and projects at the same time. So because there are so many different things that you can do with Odin, I'm actually going to walk through 3 short stories here. So first I'll talk about the kind of bread and butter application of characterizing cell phenotypes. And then I'll talk about how you can look at growth and metabolism at the same time and what that can tell you about your cells. And then finally I'll talk about how you can use this very same technology to actually identify unknown microbes in the environment. So for the first story, characterizing phenotypes of any cell. So hopefully this is super obvious, that the link between a genotype and a phenotype is very complex. So we've known for a long time that this is not a one-to-one relationship, and if you think about it, phenotypic information actually offers a more comprehensive view of the really meaningful and functional traits of an organism. So the genotype won't tell you what's actually happening in the real world, out in the wild, if a microbe is growing in the presence of certain carbon sources, or in the presence of other species of microbes that are competing for those carbon sources. And if you're trying to harness microbes to make a product, or if you're trying to fight them to protect human health, knowing how they behave is always going to be what's more important than just sequencing them. And so this shows the breadth of our panels that we call phenotype microarrays. So we have this comprehensive set. In this case, there's 20 of these plates that are specifically designed for profiling microbes. And you can probe many different pathways at once. So here we have substrates that are specific carbon sources, specific nitrogen sources. There are inhibitors like antibiotics or antifungal compounds. And then sometimes there's combinations of conditions, such as various amino acids at low pH, like you saw in the fusarium example. So, this set of 20 plates, for example, it looks like a large, overwhelming set, but that's really where Odin comes in, because it's really quick and straightforward to do this experimental setup. These plates are all pre-filled with all of these conditions, and so you just inoculate your culture into every well, stick them in the instrument, and let it go. And since there's room for up to 50 plates, that means you can measure up to 4,800 conditions at the same time, which is a really powerful way to thoroughly characterize an organism or a cell, or to make comparisons between 2 or more different strains. And as I mentioned, this phenotypic characterization is very complementary and different to genetic profiling. So this really helps you assess the fitness or the robustness of a strain or an organism. So for each one of the thousands of conditions on the previous slide, Odin automatically generates these kinetic curves and allows you to compare them. So it's really quick to see, for example, which conditions show the same behavior, for 2 different cell types, because the curves would overlay right on top of each other. But in this example, you can see that culture B in yellow had a shorter lag time on this particular substrate. So it was a bit better suited to grow on this food source, but both cultures eventually reached the same endpoint after some time. And so this would be one well out of thousands of wells that you would generate these comparisons for. And here this is exactly, the same story, but instead of a cartoon graph, I'm showing some real data that we're collecting on Odin. So for these 2 different substrates, on the left we've got substrate A, on the right substrate B, we grew 2 different strains of E. coli. So one was a wild type lab strain and one was a mutant strain. And so for substrate A, you can see that both strains are able to respire equally well. Those curves overlay quite nicely. We could measure the rate, and there's essentially no difference. For substrate B, however, the mutant strain in blue shows a much shorter lag phase and quite a different shape of curve. So if you only measured the plate, once or twice in a plate reader, instead of doing these kinetic measurements, you would miss this difference in behavior, and you might assume that both of the strains had equal fitness on this substrate. But of course, these kinds of differences can have really big implications if you're trying to model the growth of an engineered strain, or if you're trying to figure out which species will outcompete another in a community of microbes. So if you're making that pairwise comparison between two strains or two cell lines, Odin does all of the analysis automatically. So you would tell the software which sample is your reference sample, which in this case was our wild type E. coli strain, and then which sample is your test or experiment sample, which in this case was a strain that had a deletion of the MALF gene, which is part of the maltose transport system that is required for the bug to be able to use maltose or other dextrin and starch-derived sugars as carbon sources. So here I'm just showing one well in this experiment that confirms that, yes, I'm not able to see metabolism of the light blue curve in this well compared to the pink reference sample. So this tells me that deletion of the MALF gene is sufficient to see the loss of function for this strain to be able to use maltose. So as opposed to the reference wild type strain, I don't see this growth. But that was just one well. So because we're looking at so many conditions at the same time, Odin makes it really easy to find other phenotypes that we might or might not expect. So here on the right, we can see those maltose curves that I showed before, along with maltotriose, which shows the same loss of function for the mutant. But on the left side, you can see a gain of function for resistance to some tetracycline antibiotics, which also makes sense to us in this particular experiment because there was actually a chlorotetracycline resistance marker that was inserted at the MALF locus via a transposable element. So if you didn't know what to expect in terms of what genetic differences had been made, either intentionally or maybe through random mutagenesis, for example, this is a very fast way to be able to screen through many conditions at once and quickly identify significant differences between any pair of strains. And many of these pathways are interconnected, and so even if you think you know what effect a mutation will have, it's really nice to be able to screen the conditions at the same time so you can identify what's actually happened. So, the outliers on this scatter plot, there's a few that I've highlighted, which are the expected ones, but you can see there's some other gray dots that are outliers, and those might be things that we didn't expect that happened as a result of this, of creating this strain. And so just before we move to the second story, I wanted to show a quick screenshot from Odin's software, which makes it very fast and easy to collect and to see this data and even export it with just one click. So Odin knows which compounds are found in each well of these plates. And so it quickly just generates these kinetic curves, and it gives you a report with all of the significant changes between your strains for you. So it really saves time in doing the analysis as well. So now that I've convinced you how important it is to get this phenotype of your cells, I wanted to talk about another thing you can do on Odin, which is to monitor growth and respiration at the same time. So now that you're used to looking at these colorful kinetic graphs, this one includes our redox reporter dye like before, And so we're measuring metabolism, and we're making the same comparison between those two strains. This is the same thing I showed you before. But if we do the same experiment and we don't add dye to this plate, we can measure the turbidity or the growth of the cells. And so there are cases in which an organism can undergo metabolic shifts without changing their growth rate. So for example, there's studies done in fungi that show different cell phases can have different metabolic profiles, even though they maintain the same doubling time. And then the inverse can be true as well. So sometimes a specific inhibitor or stress means that growth is impacted, but metabolism and NADPH production stays the same. So microbes sometimes have preferred food sources for, cell wall biosynthesis, for example, and so they might not grow even though they're able to respire and stay alive. So these are the two different measurements that we can make at the same time in Odin. And so again, here, both of these curves are from those two strains of E. coli, and now we're just looking at one well on one of the PM plates. And you can see that for this substrate, the wild type strain shows more growth in the gray on the left, but on the right, the two strains show a very similar pattern of metabolism. And so we're seeing one strain is just persisting, not really growing, but they're both able to respire. And so depending on what property you are trying to select for or you're looking for, it's really nice to be able to collect all of this data and distinguish these types of differences on a single instrument in one experiment. And so here now I'm showing a whole plate. This happens to be number 11 in our series of PM plates. And this particular plate has inhibitors in series at increasing concentration. So as you can see in the bottom, there's these groups of 4, and you go up in concentration. So on the top, I'm showing the plate grown with no dye, and on the bottom, we use dye to measure the metabolic activity. And you can see that with the dye, on the bottom graph, the two lower concentrations of the drug, this happens to be a DNA topoisomerase called enoxacin. There's basically no differences there. So you can see that you have that dark blue color which is overlaying the two samples. So there's basically no differences in those two. And then at the two higher concentrations of the drug, neither strain is able to survive. So those basically look the same. But then when I run without dye on the top plate, I can see that the pathogenic strain is not actually actively growing. It's basically just using its energy to stay alive or persist. And then again, in the two higher concentrations, neither strain is able to grow. But you can really see that clear difference between where you see the pink curves without dye. So this is a really interesting difference that you can tease apart by doing this type of experiment. And this is super useful, as you can probably imagine, if you're trying to optimize a fermentation process. So here on the left is our set of 20 PM plates again. And you can see, each well basically probes different questions. So you can interrogate microbes to see which ones are more sensitive to high salt conditions, which ones are more sensitive to heavy metals, different pH. And so this really helps you make these calls about how fit or how robust a strain is under a given set of conditions. And on the right, I'm showing another one of the PM plates, this time, with the same two strains as before, but just a different set of compounds. And you can see that the mutant strain here has actually lost the ability to metabolize serine and sorbitol, among other things, but it has an improved ability to metabolize sucrose, for example. So you can see, different food sources that it prefers. And I'll just mention too that this set of plates is our standard set of panels that we produce, but we can also work with you to customize plates. So we do this, for particular applications or conditions. So if you're interested in a specific set, we can work with you for custom plates. So when you measure both growth and respiration in one experiment on Odin, what does this tell you? And again, it really comes back to what the cells are eating. So if you're feeding the organisms something suboptimal, then they will be in a stressed condition and they won't behave the same as when they're in an environment that is not deprived of their optimal nutrients. And you don't have to run a single pure culture on our plates. We have a lot of really interesting applications where people study whole communities and get phenotypic profiles of the community. And when you do that, you can tell, who outcompetes other organisms. So it's super interesting if you have bacterial respiration competing with fungi, for example. So you can do that to see who is eating and how that community changes under different conditions. And then finally, if you're trying to make a product through fermentation, you might really care a lot about making sure that it's respiring more or growing more. So if you're making beer, for example, you don't want the yeast to increase too much in biomass, which might foul your batch. But if you're making some high protein foods like yogurt or meat substitutes, then you do want a more biomass-rich fermentation. And so getting this really comprehensive characterization of your strains or your organisms can be very helpful for bioprocess improvements. And when people say select the best production strain, that can obviously mean a lot of different things. So the best strain because it can use the most cost-effective substrates, or the best strain for an environment where you have certain substrates and not others, or whatever the conditions that you're trying to select for. So you can use these profiles to help you optimize all of that before you scale up your fermentation. And I didn't talk specifically about this today, but you can also use this profile as a kind of a fingerprint to make sure that you have the bugs that you think you have in your fermentation, and importantly, that they aren't changing too much after dividing for many generations. So you can use this to kind of keep checking and making sure that nothing has drifted too much. And with that, I have one more short story about how you can use the same chemistry, same concept, to identify unknown microbes. So here I'm showing one of our identification plates, which is a single universal test panel, and this particular one can identify both gram-negative and gram-positive bacteria. And the way this works is there's a unique substrate or chemical in each well of the plate, just like the other plates I've shown you. And I just take, an unknown microbe that I've cultured or I've streaked out on an agar plate. I dilute it in Biolog's minimal media, and then I just aliquot it in all the wells of this plate. And that really takes about a minute to do. Then you just incubate it in Odin. And for each well, if the organism is able to utilize that substrate and respire, the dye will turn purple. And some of the wells have inhibitors in them, so if the organism can survive in the presence of those inhibitors, you will also see the dye turn purple. And these plates are designed in such a way that each organism at the species level, and sometimes even at the subspecies or strain level, will actually show a unique pattern on this combination of compounds. And so that's essentially like a fingerprint for that organism. So Odin reads the plate, and then at each interval it takes that pattern and it tries to match with something in our database, and it just keeps going until it's able to try to find a match. And so what's in our database? We have over 1,500 species of aerobic bacteria. There's 361 anaerobic bacteria. And then we have a bunch of different yeast species and over 700 filamentous fungi and molds. And this is a really powerful database because of its size and also because of its relevance. So, some of these fungi in particular are very difficult to identify with other methods. And we have a large number that are really common in the environment, like in the air and in food. So to use this kind of microbial ID for environmental monitoring, all you have to do is take swabs or swipes from, it could be different areas in your manufacturing suite, or it could be your raw materials, or even your final food products, to be able to identify and then, of course, mitigate contaminants really quickly and easily. So this is a very easy experimental setup, and it's using the same concept, but to do identification. And so we have 4 different test panels that are for different types of organisms. So we have one for yeast, one for fungi, one for anaerobic bacteria, as well as the one that I showed you. And so we've used these for quite a while with our other instruments. So for this particular proof of concept, I'm just showing a table where we took all 4 types of our ID panels and we ran them with a variety of different microbes on Odin. And we were indeed able to identify this wide range of species, just as we expected. So this just shows that, we were able to capture this on Odin as we have before on our other instruments. And so this method is a really complementary way to other ways of identifying microbes. And this uses our large database. Like I mentioned, if you add up all of the different strains, it's over 2,900 in total. And we are always expanding the list of species and strains within the public Biolog database. And if you're working with novel proprietary strains, we can work with you to make a custom database as well. And then I should also mention that our software and protocols can be CGMP compliant. So with that, hopefully you've seen how Odin can make life easier and give you lots of rich phenotype data on your strains or your cells. And, different from a plate reader where you're basically going to perturb your cell's growth every time you take them out of an incubator to read them. And you'll also miss important data that happens in between those reads, like you saw in some of those kinetic curves where, based on the pattern of the curve, you can miss a lot. So you can use this dedicated instrument that goes with our user-friendly software and the plates that all really work together to give you this very robust and actionable data. And I'll just end by showing that we have a whole ecosystem to help our researchers characterize cells, and these can be both microbial and mammalian cells, and also for identifying microbes phenotypically. And we do this with our fit-for-purpose plates and the chemistry that I've talked about today. So Odin is the brand new 50-plate version of our instrument, but we also have a smaller instrument called the MicroStation that can read 1 plate at a time and can do microbial identification. So our ecosystem also includes sample prep help. We have a lot of different protocols, and we have training and support from our expert technical support team. And then finally, we have a lab service and testing facility that is ISO certified. So as well as the phenotypic characterization and ID that I talked about today, that group can also do genomic sequencing, MALDI-TOF, enumeration, and other types of lab services that are super relevant to anyone who's working with microbes. So thank you so much for your time today. I've included our email address in case we don't have time to address your question live, but hopefully we will. And I really look forward to hearing what new and exciting things that researchers do with this very newest addition to our Biolog family.
Thank you, Dina, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started here. All right. Our first question is, Does Odin require an internet connection to do microbial identification?
Hi, thanks for the question. No, actually. So with Odin, I mentioned this large database that we have, and it's actually locally stored on the instrument computer. And so you don't need to be connected to the instrument. When you measure one of the ID plates, Odin's just searching the database that's local, and it can make a match. So if you have restrictions around connecting to the internet, no, you don't need an internet connection.
Wonderful. Thank you. I'll go ahead and move on to our next question here. What is the temperature range that this instrument can incubate? Is there a limit to how long an experiment can be?
Yeah, great question. So to accommodate, a wide range of different organisms, Odin can incubate from 22 degrees up to 45 degrees. So that covers pretty much any, type of cell line or microbe. And, there are some really slow growing fungi, as I'm sure you're aware. So Odin can just keep going for days and days and just keep incubating and keep measuring. So there's really, there's no real limit in terms of if you have slow growing organisms or very fast growing organisms, then it kind of accommodates all sorts of cells.
Perfect, thank you. And just as a reminder, we have that ask a question box in the lower left corner. Type out any questions you have from the presentation and we can do the Q&A live with Dina here today. Our next question that we have here, how can you measure multiple species at the same time?
Yeah, so we have a dedicated plate that is sort of specifically designed for studying communities, but you can really use any of our plates to look at a community of organisms. And so one thing that a lot of people have done, and there's lots of examples in the literature that show you can take your community and you just the same way you just inoculate the same community on every well of a plate. And people have looked at how that community changes in response to different, environmental conditions or over time. And so you're essentially measuring the whole respiration or growth of the whole community. So you're looking at really change over time, which is it's a really neat way to do that without having to purify them and culture them separately. So like I said, there's lots of interesting papers of people studying, wastewater or lake water, and what happens, when there's certain contaminants in that water and how that changes when they clean it up or when it gets worse.
Great. We have a question here. Can you explain how exactly the respiration rate is determined from the NADPH or NADH levels?
Yeah, so, I mentioned we have this dye and it is colorless when it's oxidized. And then as it gets reduced, just because it's in the presence of these reactions, the dye changes to, different shades of purple. And that's an irreversible reaction. So once it's purple, we can maintain that color. And we're really just measuring, the color in Odin. And so you can quantify that and you can compare it and you can look at how that changes over time. And, if you're interested, because this technology at Biolog has been around for a long time, there's a lot of papers and we also have a lot of material on our website or other material we can send you on how people have used this and how the chemistry works in more detail. But that's really essentially it. You're looking at the color change and you're quantifying that. And then at the same time, you can measure without dye, like I said. Right?
Great, thank you. Our next question here that we have is, can I find out whether certain compounds are in your plates?
Yes, of course. So we do have plate maps with, all the different panels that we make. And like I said, that can be useful if you're trying to customize plates, just to know what we have available. So yeah, check our website or reach out to us at this email address because we can share, what's in our existing plates. And if there's something that you don't see, then please talk to us.
Great. All right. It looks like we have one more question here in the queue. So if you have any questions that you'd like to ask our speaker today during the live, please do so now. I'll read out this last question we have here. What we would like to do some clustering analysis with this type of data. Is it possible to export the data?
Yes, it's actually really easy to export the data. So we've made it essentially a one-click button where you can export all of the raw data. So that would be all of the absorbance reads at every time point. And we can do that in a couple of different formats. So depending on what type of scripts or application you're using to do your analysis, you can get it in a couple of different really convenient formats that make it easy, because we know there's people who like to take all that data and then do their own treatment of it. So we've tried to make that very streamlined.
Okay, great. It looks like that was all the questions that we had come in so far. Right. So we want to, thank you for your time and your important research today. Oh, we just got one. So let's ask that one before we close out. Can we measure growth or even respiration using our own plates and media? Thank you for the question.
So for Odin, we, let me see, what is this? Using our own plates and media. So why don't you reach out to us to talk about that? Because, we do have sort of certain plate formats and certain requirements that we're using. And to be able to do that properly, it usually has to be in a Biolog plate. So, but, if there's something that you're trying to do, we'd love to find out more about it and chat with you. So reach out and see, what's possible there.
Perfect. Okay, then. So thank you for your time today and for your important research. We would also like to thank LabRoots and our sponsor Biolog for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions that are submitted during the on-demand period or those that we did not have an answer for will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you everyone, and goodbye.